Okay, good morning, everyone, and welcome to the 2022 edition of the Royal Tyrrell Museum Speaker Series. My name is Francois Terrier, and I'll be the host today. So again, this year, due to the COVID-19 pandemic, the Royal Tyrrell Museum Speaker Series will be live streamed on Zoom as a series of weekly webinars. This technological leap is made possible thanks to the generous support of the Royal Tyrrell Museum Cooperating Society. At any time during the seminar, if you have questions, please use the Q&A box or button at the bottom of the screen so you can type your question, and then after the seminar, we'll go over them with the speaker to answer your questions. So to kick off the 17th season of the speaker series, the Royal Tyrrell Museum and its cooperating society are proud to present Dr. Bence Viola. Bence is a professor in the Department of Anthropology at the University of Toronto in Ontario, Canada. Bence was born in Hungary but grew up in Vienna, Austria. He received a master's degree from the University of Austria for his study on the link between the internal structure of the femur and locomotion in primates. He subsequently moved to France to pursue a master's degree at the University de Bordeaux, too, where he studied Miocene apes. And then finally, he returned to the University of Vienna to pursue a PhD on the Neanderthals from Central Asia. Following his PhD, Bence spent 5 years at the Max Planck Institute for Evolutionary Anthropology in Leipzig, Germany, as a postdoctoral fellow before moving to the University of Toronto 7 years ago. Bence's research focuses on better understanding the cultural and biological interactions between archaic human populations from Central Asia and Europe. And he is also interested in the evolution of bipedalism, biomechanics, and the structure of the inner ear. Over the years, he has conducted extensive fieldwork in Central and Northern Asia, as well as in Central Europe. So today, Bence will present an overview of his research on the different extinct human species that inhabited Central Asia during the late Paleocene. That would be quite a discovery, the late Pleistocene. So without further delay, I'll introduce you to Dr. Bence Viola. So Bence, the computer is yours.
Yeah, thank you, Francois. Thank you very much for the invitation, and of course, thank you all attendees for your attention and your presence. I'm very happy to be able to speak about my research here shortly. I will just start. I'll just share my screen so we can get started with this. So as Francois mentioned, my interest is mostly in what was going on in a region that was relatively poorly known for a long time, Central and Northern Asia. Since a long time, we've known that humans had extinct relatives. This is well known since the 19th century. The first extinct human species that was discovered were the Neanderthals. These here are the remains of an individual who was found in the Neander Valley in Germany near Düsseldorf, after which valley, the Neander Valley, the Neanderthals, this whole group is named. They are probably the best known fossil hominins. We have fragments of several hundred individuals, but actually only a few reasonably complete skeletons. We have a reasonable idea of how they looked, even though it's actually fascinating how our views of Neanderthals have changed over time. We reconstruct them quite differently depending on when these reconstructions happen and with what kind of approach the artist goes into it. I think that these days we tend to look at them as much more similar to us than people thought so in the early 20th century, for example. For a long time, Neanderthals were thought to be a primarily European species, but it became clear that they were also relatively widespread in the Near East. And since the 1930s, we've known that they also existed in Central Asia. Why am I interested in Central Asia? Central Asia is really an extreme in many ways. As you can see here, we actually have a few sites, but we'll look at them more in detail. Central Asia is a large region that is characterized by climatic extremes. It contains some of the most extreme deserts in the world, for example, the Taklamakan and the Gobi. It contains gigantic mountain ranges, such as the Himalayas and their outskirts, the Pamir and Tian Shan. It also includes more moderate mountain ranges like the Altai. It's connected or towards its center. It also contains the steppe belt that connects Europe and East and Northern Asia, which likely was an important migration corridor and is, in my opinion, one of the reasons why actually Neanderthals, the species that was more typical in Europe, actually came this far. Once you adapted to the environment of these steppes in Eastern Europe, it was relatively easy to move in the same ecozone quite a bit further east. The situation was more complicated in reality, of course, because the steppe zone was not stable the whole time. We know that, for example, large parts of Western Siberia were actually covered by large swamps and lakes during different periods of the late Pleistocene, which might have hindered movement quite a bit. And of course, towards the north, this vast region is delimited by the boreal forests of Siberia. And honestly, we have very little idea of what was going on up there. Fossil preservation in these regions is not very good. And of course, due to the relatively scarce settlement of the area, there's also much less archaeological activity than one would wish. So we're looking at a large and, in the end, even though ecologically diverse, but also in many ways quite extreme region. And looking for fossil hominins in this area is not a completely new idea. Actually, one of the first major interdisciplinary paleontological expeditions, the Central Asiatic expeditions of Roy Chapman Andrews, in the 1920s, actually went into Central Asia, into Inner Mongolia and the Gobi Desert. Roy Chapman Andrews thought he would find some human ancestors there. He was not successful, but of course, as you know, he found other things that were just as interesting. Of course, the immensely rich treasures of dinosaurs and early mammals in the Gobi. In 1938, Alexander Okhladnikov, a Russian archaeologist, started excavating various sites in Central Asia. And the most important discovery that he made was in 1938, where in the cave of Tashiktash, which is in what is Uzbekistan today, at the time, of course, part of the Soviet Union, he found a partial skeleton of about 10 or 8 to 12-year-old kid, most likely a boy, the so-called Tashiktash child. Okhladnikov and his colleagues recognized quite quickly that this looks very similar to the Neanderthals that were known at the time from Europe and the Near East. And so Tashiktash for a long time was the easternmost Neanderthal. Interestingly, very little other human remains were discovered for more than 50 years. But since the 1980s, my Russian colleagues from the Institute of Archaeology and Anthropology of the Siberian branch of the Russian Academy of Sciences in Novosibirsk have been working in the region where I want to take you now, the Altai Mountains of Siberia. So as you can see, we're here in Siberia, but Siberia is not all an Arctic wasteland. Most people, think about Siberia and think it's like the far north. Denisova Cave, for example, the most important site in the region, is roughly the same latitude as Leipzig, or actually south of Berlin. In Canada, it's a little bit further north, about the same latitude as Calgary, but that's not far from the Arctic. So in the in the late 18th, early 19th century, a hermit named Denis lived supposedly in this cave here. In this picture, you can see I'm standing actually above the cave and look down into the valley of the Anui River, a tributary of the Ob, that then, with the Ob, of course, goes into the Arctic Ocean. We're in the foothills of the Altai here, which is really a medium mountain range. Denisova Cave is at about 750 meters altitude. The mountains around here go up to maybe 1,300 meters. If you go about 100 kilometers, you get into actually high altitude with 4,000 meter high peaks. But most of the mountain range is actually quite low. These days, it's mostly a mixed forest, some deciduous trees, but lots and lots of firs and pine trees as well. It was likely quite similar in the past as well. And the valley here was actually important because it's a useful migration corridor for fauna between the pastures and open meadows in the mountains and the steppes that are further north. So since the 1980s, my colleagues are working here, and they actually built a little scientific camp here with houses for visiting researchers, for the students excavating there. And they excavate 3 to 4 months each year since the 1980s. So the very large amount of material has been excavated from this cave. This here is the entrance to the Denisova cave. As you can see, there's actually 2 entrances. There's the main chamber on the left, the large entrance, and on the right, there's a smaller entrance that takes you into the so-called South Gallery, which was not excavated for a long time, but current excavations are actually focusing on that area. I'm not going to go into details with the stratigraphy in Denisova. I'll just say it's rather complicated. Caves in general tend to have complicated stratigraphies because you have lots of activities, sediment moving around due to water, due to gravitation, due to gravity. And in the case of Denisova, also due to the activity of various carnivores. Besides human occupation, the cave was also inhabited by numerous carnivores, primarily hyenas and cave bears, who dug around quite a bit and moved things around, which of course messes up everything. Another problem is that the excavations in the 3 different parts of the cave, the main chamber, the East Gallery, and the South Gallery, are independent from each other. They're not connected stratigraphically, and they've only been correlated based on faunal and geological data, and so it can be a little hard to compare these things. Originally, it was thought that the numbering, like the layers, like layer 9 in the main chamber, the East Gallery, and the South Gallery, is equivalent. We know today that this is not true. For example, things that are layer 13 and 14 in the East Gallery are likely contemporaneous with like layers 19 and 20 in the main chamber even. It's a bit of a mess to be honest, and of course the somewhat limited documentation from the early parts of the excavation is not very helpful either. In the last 5 years, there's been a very large dating program working with colleagues from Australia, the UK, and elsewhere, trying to figure out the exact dating of the sediments using optically stimulated luminescence and C14. Additionally, we also have data from DNA. I'll talk about it a little bit later, but we can actually use ancient DNA to very tentatively, at least, assess the age of fossils that they come from. In general, it seems that most of the occupation of the cave happened in the last, depending if you accept very early OSL dates, it could date back to more than 300,000 years. I'm a little skeptical. This does not fit very well with the fauna. The faunal material for these early layers fits better with less than 200,000 years ago, but I'm not a micromammal specialist. I just have to believe what my colleagues tell me. But it seems that most of the deposits come from the last 200,000 years. Denisova Cave is mostly known for one thing: this tiny little speck of bone that you see here. You're probably used to dinosaurs, spectacular large fossils. In paleoanthropology, we have to work with much more limited material. This here is the fragment of a finger bone, of a terminal phalanx of a juvenile individual. We know from genetics that it was a female, it was a young girl. who died in this cave somewhere between 60,000 and 100,000 years ago. And as you can see, it's a tiny little piece. Morphologically, there's very little we can say about it. We have here micro CT based renderings, and as you can see, actually, this consists of two parts. In blue, you have the epiphyses, and in green, the diaphyses, so the shaft of the bone and the articular surface, which haven't been fused completely yet, indicating that this was a juvenile individual. In reality, when this fossil was originally found, it was actually a complete phalanx, but it was cut in two, and sadly, the photographs that were taken before it being cut in two are lost, which is really tragic because we only have very tentative reconstructions of how this fossil might have looked originally. But we also have some other fossils. Again, compared to dinosaurs, not very spectacular, but this is a beautiful tooth. I think it's probably the, and I'm biased, but it seems like one of the most beautiful teeth in the world to me. It's a very large molar, likely an upper second or third molar. And so this was, I'll get to the DNA in a moment, but when the finger bone was discovered and analyzed, we realized that there might be something in this cave that is neither a Neanderthal nor a modern human. And in 2010, I traveled with my geneticist colleagues to Siberia to tell our Russian colleagues about this. And one of them pulled this tooth out of his shirt pocket and showed it to me. And the sheer size, the massive roots, the complex occlusal surface, and even the shape of the crown made it very clear to me that this is true. The geneticists are not making up something. Clearly, we're looking at something that is neither a Neanderthal nor a modern human here. Because the material we had at the time was extremely limited, as I showed you, that tiny piece of the finger bone and this tooth, we had to use other methods to better understand where these fossils belong phylogenetically. And we were very lucky because over the course of the 1990s and the early 2000s, a new field, ancient DNA analysis, emerged. I'll give you just a very, very short introduction to this because it is very important for the story of Neanderthals and Denisovans in Central Asia. In the 1980s, it became clear that DNA can, under certain circumstances, survive for remarkable amounts of time. The first studies were looking at museum specimens of, for example, of the quagga, relative of zebras that went extinct in the 19th century, or at mummies. And people showed that there's actually some DNA preserved after hundreds, and in some cases, even after thousands of years. People, of course, then asked themselves, how far can we go back with that? And by the early 2000s, and by 2010 for sure, we knew that up to about 100,000 years ago, you can actually relatively reliably extract DNA. So how do we do this? And this is a very, very simplified workflow. The first thing is you have to take a sample from your bone, which of course means that this is a destructive method, which is one of the things why paleoanthropologists, looking at the very limited fossil record of humans, of course, are usually relatively careful to do this. In the beginning, very large samples were taken. These days, luckily, we can work with very limited material. You take a bone sample, then you use various chemicals that destroy amino acids, that remove inorganic parts, and so on, and really just separate out the DNA in the sample. Then you amplify the DNA, so you create many, many copies of the DNA, and then you sequence it. You read out the sequence of the bases of which the DNA is composed. Now, this sounds very easy. In reality, of course, it's very hard, especially when you're working with old material. With anything that is pre-Holocene, I would say, there may be 4 or 5 labs in the world that are able to do this, and 1 or 2 that are reliably able to do this. But it actually works. There are quite a few limitations, though, of course. If you think about it, in recent bones or in recent tissue, you have usually about 1 microgram of DNA per gram of tissue. And the DNA comes in long strands that are then coiled into chromosomes that build up our genome. And if you think about it, the modern, our present day genome consists of about 3 billion base pairs. and it's only 1 microgram per gram of tissue. Of course, when you work with DNA, you always have to keep in mind, because DNA is in every kind of living matter, contamination is always an issue. But when you work with recent DNA, usually the contamination is less than the actual DNA you're working with. Ancient DNA has a few, let's say, annoying traits. One of them is that you have much, much less DNA than in present day tissues. Usually between 10,000-fold less up to 1,000,000-fold less. So you're working with much, much less material. And also, what happens is that the DNA doesn't come in these long strands anymore. It breaks up into tiny pieces. As I mentioned, the recent human genome is about 3 billion base pairs. When you're working with ancient DNA, the genome breaks up into tiny pieces of usually less than 50 base pairs, so 50 letters of the genome. You have to imagine this more or less like you take a book, and it's very easy to read it when it's a complete book. But if you take this book, you put it through a shredder, and then try to read the book based on just the shreds that come out, it's going to be very hard. But you can actually work around is as long as you can actually get out fragments that have a certain length, and the cutoff is usually about 30 to 32 base pairs. You can use statistical and computer methods to actually reassemble the genome from those fragments. You can just try to overlap them. But there's some other things too. One of them is that ancient DNA also undergoes damage, and the damage is quite characteristic. It usually happens close to the ends of these fragments, where certain bases change. And we know quite exactly that what kind of changes happen. And so we can actually use this today to authenticate tissue. So the damage is in one part annoying because it can give you false results. But on the other hand, it's helpful because we know that this damage only happens after a certain amount of time. And so you can actually use the damage to separate old DNA from recent DNA, so from contamination. Another problem that is very big with, especially if you try to get ancient DNA from bones, is that you also have non-endogenous DNA. And so this is DNA from bacteria and fungi that are in the bone. And in many cases, this non-endogenous DNA can actually be much more than the Neanderthal or some kind of human DNA that you're looking for. It can be like almost 100 times more. We have some specimens where less than 1% of the DNA is actually from the individual we are studying. And then, of course, contamination, which in the case of recent DNA is a small problem, becomes a much larger problem. Because if you have the same amount of contamination, but you only have, 100,000-fold less original DNA, then the contamination becomes a bigger issue. Amazingly, we can work around all these things. Svante Pääbo and his team in Leipzig, where I did my postdoc, spent the last close to 30 years working on this issue, and they actually managed to get around most of these things. So we can actually extract the DNA, and we can study the DNA of these fossils. So in 2009, Johannes Krause, who at the time was a PhD student in Svante's lab in Leipzig, looked at this finger bone from Denisova cave. And what he found, looking at the mitochondrial DNA, and mitochondrial DNA is just a small part of our genome that is in the mitochondria and these cellular organelles in our cells. He looked at mitochondrial DNA and found that the mitochondrial DNA of this finger bone looked very different from what we see in Neanderthals, but also in modern humans. So if we use the molecular clock to reconstruct when the last common ancestor of chimpanzees and modern humans lived, and we set that at, let's say, 7 to 11 million years, then by this time we already had mitochondrial DNA from Neanderthals. Neanderthals and modern humans split about 500,000 years ago. What Johannes noticed is that this finger bone from Denisova cave seemed to be much more different, actually twice as different than Neanderthals and modern humans. And so it likely indicated that this was a different population, a group that we didn't know about before. And so because we didn't want to name a new species, we actually said we'll just call them just like Neanderthals, or name Neanderthals after the site where they were found. We'll call them the Denisovans after Denisova cave. Now, Mitochondrial DNA has a few characteristics why it's quite popular with when you work with ancient DNA. One of them is that in each cell you only have two copies of the nuclear genome. I mean, one copy, but it's twice, because each chromosome exists twice. But you have thousands of copies of the mitochondrial genome. And also mitochondrial DNA is much shorter. The mitochondrial genome is about 16,000 something base pairs instead of 3 billion. So it's easier to reconstruct. Mitochondrial DNA is easy to study, but it has one relatively unpleasant characteristic as well, and that mitochondrial DNA is matrilinear. It's inherited only via the maternal line. So you get it from your mother, she got it from her mother, she got it from her mother. So if you go back in time with mitochondrial DNA, you're only looking at a very, very limited part of your ancestry. Unlike with nuclear DNA, your nuclear genome, that actually comes from both of your parents and from all your grandparents and great-grandparents and so on. Interestingly, when we looked at the nuclear DNA of this finger bone from Denisova cave, we found that the phylogeny we get from it is not the same as the one for mitochondrial DNA. Actually, if you look at the nuclear DNA, you find that Denisovans, so the finger bone, and Neanderthals are more closely related than either one is related to us. That was quite a surprise for us. Because this then indicated that Denisovans are simply more or less the Asian cousins of Neanderthals. One thing that is great about nuclear DNA is that actually you can look at ancestry in much more detail because your nuclear DNA comes from both of your parents and they got it from both of their parents. Your nuclear DNA actually samples your complete ancestry. And of course, it also samples the population that you come from. So we can look at things, for example, we can look in much more detail at how these populations are related to present-day humans. So we knew we have a population that was previously unknown, the Denisovans, but we wanted to understand how they are related to present-day populations. The method we used here is usually referred to as Patterson's D or Abababa. I'm not going to detail the statistics. What you do is you compare an archaic genome to two modern humans, and you check to which one it's more similar. And theoretically, if you're looking, for example, African and European populations are equally similar, are equidistant from Neanderthals, you'd expect that in half the cases, the Neanderthal is similar to the Europeans, and in half the cases, it's similar to the Africans. What you actually find when you do this is that Neanderthals are a little bit more similar to modern humans. To modern Europeans than they are to modern Africans. And they're also a bit more similar to modern Asians than they are to modern Africans. And they're a bit more similar to modern Papuans or Australians than to modern Africans. So we took more or less 5 modern humans, just to keep it very simple, kind of looking at different ends of the migration out of Africa. We know that, the ancestors of modern humans lived in Africa and they moved out somewhere between 50 and 100,000 years ago and colonized Europe, Asia, Melanesia, and then, of course, later North Africa, the Americas as well. When we compared the Neanderthal genome that we had to all these modern humans, what we saw that Africans are about as far from Neanderthals as you get. But Europeans, Asians, and Melanesian populations all contain a little bit more Neanderthal DNA than African populations. And what we interpreted this as was simply that likely when modern humans moved out of Africa, they met Neanderthals somewhere and they interbred with them. And populations, as they later expanded, carried this little bit of Neanderthal ancestry with them. And it's little, it's 1 to 3%. But, 1 to 3% is still, it's not nothing. We did then the same analysis with the Denisovans. And just to remind you again on the map, Denisova cave is here more or less in the center of the map in Central Asia. We, of course, didn't find, because you have roughly the same amount of Neanderthal DNA in modern human populations outside of Africa, no matter where they are. We think that maybe this happened quite early once modern humans moved out of Africa, but it's not 100% sure. It might have been more complex as well. So we then looked at Denisovan DNA, and we found that there's, no Denisovan DNA in African populations. There's nothing in European populations. We found that there's a tiny bit in Asian populations. But what was really surprising is that actually populations in present-day Melanesia, in New Guinea and Australia, actually carry relatively large amounts of Denisovan DNA, between 4 and 6%. And this is in addition to the Neanderthal DNA that they already carry. This was quite surprising. The first individual that we got this signal from actually came from New Britain, which is an island of New Guinea. And when I was first told this, I was like, this is ridiculous. This is 10,000 kilometers away from Denisova Cave. Why would that be the place where we actually find Denisovan contribution to recent humans? But it's pretty clear that that this is true. We have now DNA from quite a few individuals from there, and it's clear that, or it seems that somewhere, populations that ended up in New Guinea and Australia met Denisovans and interbred with them. This, of course, likely indicates that Denisovans were not just present in the Altai, as we, at the time at least, thought. They were likely also present in Southeast Asia, for example, where these populations had to move through. Sadly, we still don't have any clear evidence for Denisovan presence there. But as you'll see later, we actually have some material that is Denisovan-like from East Asia, at least. So from all these DNA studies, what emerged was a picture of a relatively complicated web of populations that were morphologically relatively different. But in the end, whenever they came into contact, they interbred. We have evidence from DNA for gene flow between modern humans and Neanderthals, between modern humans and Denisovans, between Denisovans and Neanderthals. And we even have evidence for gene flow from what we call a potentially unknown hominin into Denisovans, which I think might be Homo erectus from Asia, which is a group that moved into Asia quite a bit earlier, more than close to 1.5 million years ago, or maybe even close to 2 million years ago. So what is fascinating is that we have these different groups present in the Altai. We actually have evidence not just for the Denisovans in the Altai, but also for Neanderthals. And I think the best evidence for Neanderthals comes from a different site, from Chigirskaya Cave, which is about 100 kilometers away from Denisova. In excavations since 2008, led originally by Sergei Markin and then later on by Ksenia Kolobova and colleagues, we recovered about 80 fragments of Neanderthals, of at least a dozen individuals. We have postcranial remains, we have teeth, we have relatively complete material as well. As you can see, for example, on the left here, there's a relatively complete right arm that seems to belong together, and it actually raises the possibility that there might have been maybe even a burial in this cave at some point. There's also a very, very rich archaeological record. Denisova Cave actually doesn't have that many lithics and not that many faunal remains with cut marks. There's compared to the amount of material that was actually excavated, the cave, at least in the earlier phases of Denisovan presence, doesn't seem to be occupied very intensively. I've worked at sites in France where Neanderthals were, where there's practically no sediment. It's like bones lying on stone tools and then bones again and stone tools. Denisova is not like that at all. There's likely much less intense human occupation. Chigirskaya, on the other hand, is like that. It's really over the last, whatever it's now, 12 years of excavations, we have about 80 human remains, but we also have several 100,000 lithics, about 300,000 faunal remains, which honestly, there's probably also some more human remains in there among those. We're currently working on finding more of them. But it seems like it was occupied very intensively. And what was interesting for me is that actually this is material that is morphologically relatively clearly Neanderthal. With the fragmentary material we had before from other sites in the Altai, where we did think that they were Neanderthals, it was very hard to tell. They were all very gracile compared to European Neanderthals, and they did not show some of the relatively typical features we see in Neanderthal teeth in Europe, for example. This material here is not at all like that. It's very clearly Neanderthal. For example, this mandible here has a very nice oblique mylohyoid line, which you see here on the right. It's this ridge that's descending from from behind the second molar. You have the so-called mid-trigonate crests, which are little crests that connect the two mesials, so the two forward cusps on these molars are connected by a little crest, which I highlighted in red here. And this is something that we see in like 96% of Neanderthals. But for example, at one of the other sites in the Altai at Okladnikov cave, it's not present, at least not on the enamel surface. Similarly, this incisor is typically Neanderthal. It shows the strongly curved outer surface. It has these ridges, the so-called shovel shape. It's very large, so it fits with the kind of Neanderthals that we know from Europe. And what is very interesting is that based on the lithic material that we have, we also think that there might be some kind of cultural connection to Eastern Europe. The lithic material we have, so the stone tools, look very similar to a technocomplex, so more or less an archaeological culture known as the Mikokian from Eastern Europe. It's known from Germany, from Poland, and then especially from Crimea. And so the stone tools we find are quite similar to this. And so we have proposed, based on the archaeology, that maybe these Neanderthals in Trigirskaya cave actually came from Eastern Europe, and relatively recently. Now, fascinatingly, Trigirskaya cave is not the only one where we have Neanderthals. We also have Neanderthals from Denisova cave, and sadly, they are not as diagnostic morphologically, but they have very good DNA. So this here is a toe bone that was found in Denisova in 2011. It's a proximal fragment of a probably third or fourth ray of a toe. Again, morphologically, it's relatively robust. It would fit with a Neanderthal, but you would not be sure that it is. But genetically, it became very clear that it's a Neanderthal. Now, what is fascinating about this individual is that we actually have a lot of DNA from it, very, very high quality DNA. And when you have lots and lots of DNA, you can do other funny things. As I mentioned before, the genome in the end samples two individuals, because your nuclear genome, in your nuclear genome, you have two copies of each chromosome, one that came from your dad and one that came from your mom. And if you have enough DNA, you can actually start working out which copy of this chromosome the fragments that you have come from. And you can actually compare the genomes, or at least the chromosomes that come from the mother and that come from the father, which tells you, for example, how much genetic diversity there is in this population. And this is important because this tells us a little bit about population size. And it also tells you how closely the parents were related. So one of the things we did is we looked at what we call the heterozygosity, and that is how similar these two copies of the chromosome are. And in these plots, which look a little bit like an ECG, like an electrocardiogram, if the red line is close to the bottom, that means that the two chromosomes are very similar. When the red line goes up and forms a little peak, that means that the chromosomes are different. And if you compare here a recent French individual, the Denisovan finger bone in the center, and then on the bottom, Altai, which is the Neanderthal from Denisova cave, you can see that the Neanderthal from Denisova cave has huge parts of this is just a single chromosome. I think this is chromosome 21, if I remember correctly. Has huge parts of this chromosome where the two parents are completely identical. This, number one, indicates that likely the population was not very diverse. But number two, it also shows that likely these parents were very closely related. And running these analyses across the whole genome, we can actually show that the possible scenarios that we're looking at are either that the parents of this individual were grandfather and granddaughter, uncle and niece, half siblings, or double first cousins, which is a little shocking. But what is important is even if we remove this recent inbreeding, the effect of the parents being so closely related, we still see that this population was likely extremely small and genetically not very diverse, which might in some way also give us a hint on why this population disappeared. When populations become very small and genetically impoverished, they of course have a hard time adapting, and of course you also have the accumulation of deleterious mutations. So you have a larger number of problematic mutations that can lead to issues later on. I mean, the inbreeding we see here is really like European royal families, like the Habsburgs would probably look similar. We can actually do this on a larger scale as well, because what we can do is we can take the genome and chop it up into little pieces and run analyses on each of those pieces to see when the last common ancestor of these various fragments lived. And when we do this, we can actually reconstruct how genetic diversity, but also population size, changed over time, which is, again, it sounds a little bit like science fiction, but you can do this. So if you do this with recent populations, the Neanderthal and Denisova here, you get a very interesting picture. So what you see here, I'm just looking, I said that I can't use my pointer to show you, but as you can see here on this graph, on the y-axis, you have population size. It's actually effective population size, which is a proxy of genetic diversity, but it's also linked to real population size. And on the x-axis, you have time, which again, is theoretically in generations, but we scaled it here roughly to age. And so you start on the right about 5 to 10 million years ago, and as you can see, all recent modern humans and Neanderthals and Denisovans seem to be equal here. There is quite a bit of genetic diversity that then starts dropping off maybe 2 or 3 million years ago, and actually decreases quite a bit, and then starts increasing again maybe 500,000 years ago. And this is when the Neanderthal and Denisovan genomes separate from us modern humans. And they go through a really strong bottleneck. Their genetic diversity and their population size really drops off. And we think that this was when Neanderthals and Denisovans, or their ancestors, together migrated out of Africa, which led to a strong decrease in genetic diversity. These genetic bottlenecks are quite well known from conservation biology, for example. And they stayed at this relatively low level till they completely dropped off, especially in the Neanderthal. Modern humans increased in population size till about 200,000 years ago, when you have a major drop. And again, this is when modern humans left Africa. And what is interesting is that this drop in population size and genetic diversity didn't only happen in the populations that left Africa, but also in a little bit in the African populations. And then, of course, over the last about 10,000 years, you have a rapid increase in recent human genetic diversity and population size. So we can get insights that we wouldn't have otherwise. We also have DNA from Chigirskaya cave, from this other Neanderthal site. And what is interesting is that the DNA from Chigirskaya, even though it's only 100 kilometers away from Okladnikov cave, is very different and is more similar to European Neanderthals. And so we actually think that the DNA indicates that the populations that came into Siberia and that we find in Denisova cave, and the population that came and that we find in Chigirskaya cave, are actually two independent Neanderthal migrations into Siberia, which again brings us back to this idea that maybe this steppe belt connecting Eastern Europe and Siberia, and then of course even further on east, might have been a place where Neanderthals could migrate relatively easily. But both of these Neanderthal populations seem to share these very low population sizes. And that is not really true for the European Neanderthals. Those seem to be a bit more diverse and better off. One of the reasons might be that simply really the Altai is the very periphery of the Neanderthal range, and these guys just were cut off from everyone else. That's one possibility. But it might also be that simply in the end, the environment was quite marginal for them. So we have this low population size. I'm not going to go into details of these figures. As I mentioned before, from the DNA, we know that you have lots and lots of contacts between these groups. But what you see in this graph is that the amount of gene flow, the percent of Neanderthal ancestry in modern humans or Denisovan ancestry in modern humans is always very low. So you would ask, why does it even matter that, we carry 1% or 3% of Neanderthal or Denisovan DNA? the fascinating thing that we found out over the last few years is that it actually matters. Even these small amounts of DNA matter. So one thing that we found, for example, is that in our immune system, we modern humans across the globe, in many cases, actually carry variants that we picked up from Denisovans and Neanderthals. And so the idea is these are the so-called HLA, the human leukocyte antigen genes. These are genes that encode various kinds of receptors that are then on white blood cells and that are used to dock onto various kinds of pathogens. And so one of the ideas is that when you go through these bottlenecks, you lose a lot of genetic diversity. As I said, the population size went down. And interbreeding with archaic humans allows you to kind of replenish the genetic diversity, which is important because a lot of genetic diversity in these HLA genes means that you can attack lots of different kinds of pathogens. The other possibility is that maybe, Neanderthals and Denisovans living in Europe and Asia and elsewhere underwent selection for certain variants that were advantageous against pathogens that were frequent there, and when modern humans came in and interbred with them, they could pick up these adaptations easily. The most fascinating thing, though, is a variant that that was discovered in Tibet. So present-day populations in Tibet carry a certain variant in the so-called EPAS1 gene, which is a gene that is linked to adaptation to high altitudes. It is known that Tibetan women who carry this certain variant have babies with a larger birth weight. And of course, babies that are larger at birth are more likely to survive, especially without, advanced medical care. And what is fascinating is that this variant that is found in Tibet, that in this so-called haplotype tree, which is an unrooted phylogenetic tree of genetic variants, is on the bottom right in pink. The variant we have in Tibet is very different from all the variants we see in all modern humans today. But fascinatingly, it's actually very similar to the variant we see in the Denisovan genome. And so the idea is that this was an adaptation that allowed that was advantageous at high altitudes. And when the ancestors of Tibetans picked this up, it became selected for very strongly, and is now present in about 80% of Tibetans. Of course, when we found this out, we asked ourselves, why would Denisovans actually carry an adaptation to high altitude? Denisova cave is at 750 meters. This is somewhat confusing. As you'll see in a moment, it actually starts to make sense later. These contacts also happen not just between modern humans and Neanderthals and Denisovans, but they also happen between Neanderthals and Denisovans. And to our utter surprise, we actually have direct evidence for this from Denisova cave. So another fragment from Denisova cave, which is probably even less spectacular than the finger bone. I think this is probably one of the ugliest fossils in the world. Denisova 11, or Deni, is this tiny little long bone fragment that was only identified because some of our colleagues are using collagen fingerprinting to identify the species of tiny bone fragments that we can then use for DNA analyses and other things. Anyway, one of our colleagues, Sam Brown, identified this tiny fragment. As you can see, it's less than 2 centimeters in length, coming from some long bone, as hominin. And when we analyzed the DNA of this individual, we found that the mitochondrial DNA was Neanderthal-like. But to our surprise, in the nuclear DNA, we found that about half the positions in the genome are Neanderthal-like and half the positions are Denisovan-like. And the best explanation for this, and we ran lots of complicated simulations and other things, the best explanation for this is that this is an individual whose mother was a Neanderthal, but whose father was a Denisovan. Which means that Neanderthals and Denisovans actually met in the Altai. And not only did they meet, but they also interbred, and they produced this one individual. And we were unbelievably lucky that we actually found a bone of this individual, who's a first-generation hybrid. when we looked more in detail at this genome, we actually found that there's actually parts of this of the father's genome, so of the Denisovan genome here, that also that are also Neanderthal-like, which indicates that likely there was already similar admixture events in the past. So the father of this individual had ancestors that already interbred with Neanderthals, maybe 10,000 years before this individual was born. And so we see here, again, this is a relatively complex phylogenetic picture. We see here two populations that separate. On the right, the Denisovans; on the left, the Neanderthals. We think that their original separation happened somewhere maybe 430, 450,000 years ago, maybe a little bit older. What is always reliable with these genetic data is the pattern, like the sequence of events, not necessarily the absolute numbers. And then somewhere maybe 100 to 80,000 years ago, these populations met again in the Altai with the Neanderthals and Denisovans interbreeding and producing Denisova 11. What is interesting is that this also supports this idea that there's two separate Neanderthal populations, because actually the Neanderthal mother of Denisova 11 is unlike the other Neanderthal from Denisova cave and more similar to the Neanderthals from Chigirskaya. So this really gives us a picture of, small populations that move around, that sometimes hybridize, but clearly also often go extinct. They just disappear, which, I think it should be a warning for us. It indicates that human populations are frail. And of course, we have much larger populations today than at the time, but extinction is never, is not impossible. It can happen to anyone. So who are these Denisovans really? And this is a very hard question. We still have very little material from them, but over the last years, research outside the Altai has really expanded our view of them. The first big surprise came from Tibet, from the region of Xiahe, where in the 1980s, a Buddhist monk found a mandible, a lower jaw, in a cave, in this cave here, in Baixia Karst Cave, which is used as kind of a sanctuary by monks. He found his mandible and took it back to the monastery, and it took about or almost 30 years to end up in the hands of archaeologists of Dongzhu Zhang from Lanzhou University, who then assembled a team who studied this material. And this is the mandible here. If you're a paleoanthropologist, you will realize that this mandible is definitely not a modern human. It doesn't really look like a Neanderthal either, but it definitely looks as something, archaic, as something pre-modern. Sadly, we don't have any DNA from this specimen, but we actually, some of our colleagues managed to extract proteins from the enamel and use that to show that this actually, or at least its dental enamel, is more Denisovan-like than Neanderthal-like. And so likely this is actually another Denisovan. And it comes from the Tibetan plateau, from relatively high up, which of course then fits with this idea that at least some Denisovans might have been altitude adapted. And so the variant we see in present-day Tibetans might come from this population. The excavations in the cave are ongoing, and there supposedly is more material that, of course, will tell us much more about the Denisovans than the, small fragments we have from Denisova cave. Even more interesting was a find that was published just a few months ago under the name of Homo longi, a new species of human from Harbin in northern China. This specimen again has not a lot of context. It was found during bridge building works during the Japanese occupation in the 1930s. One of the workers found this cranium, took it home, buried it in a well, and then it was taken out and analyzed only a couple of years ago. Again, this specimen is clearly not a modern human. It's not a Neanderthal. Sadly, there is not much we can actually compare to the Denisovans, except the one tooth. As you can see, it preserves one of the second molars, which in some ways is reminiscent of what we see in Denisova cave. It's quite large. Similarly, we have some cranial fragments from Denisova that I'm not showing you. They are not published yet, but they likely also show a few similarities to this specimen. I'm not sure I agree of naming a new species for this homolongi, especially because some of the phylogenetic analyses actually showed that this specimen is quite similar to material that was already known from China that actually has a valid taxonomic name as well. But these are details that we don't need to worry too much about. Anyway, it seems that even in northern China, we have some evidence for the presence of Denisovans. There's actually additional material from China. There's a few teeth, for example, from a site called Chujiayao that are very similar to Denisovan. I think also belong to them. So I think we know Denisovans since just over 10 years. I think in the next 10 years we'll get a much clearer picture of who they were and how they looked. I saw before that one of the people in the audience knows much more about this Harbin cranium than I do. So maybe if he wants to, if there are any questions about that, I'd be very happy. I'd like to hear what he thinks. Modern paleoanthropology is innately teamwork. I worked with a lot of people over the last 20 years, many dear friends who were extremely helpful and supportive, and of course I would like to thank them all. My colleagues in Russia and Central Asia, and of course the geneticists who've been who are just amazing and wonderful. So I'd like to thank them. And of course, I would like to thank you for your attention.
Hey, awesome. Well, thank you, Bence, for a great talk. I think it's really fascinating seeing all the technological advances, what it allows us to do now. We're moving away from basically like classical paleontology when we're just describing bones to try to infer affinities or even relationships, whereas now going into genetic paleontology, where basically you can say so much about interrelationships and even identifying new species. So for people like me who study dinosaurs, I think those advances make us very jealous because we'll never be able to do all that work with dinosaurs. But it's amazing what can actually be learned by being able to study ancient DNA. So yeah, it's really, really fascinating. I think we have a few questions coming in, but as they come in, I have a few questions, if I may, that I'd like to ask you. One of them is, how did the bones end up in the Denisova cave? Is it a case of basically like a burial chamber? I can't remember if it's Homo naledi or another hominid that they've been proposed that maybe they brought the remains of the dead into that cave, or is it in instances you mentioned that basically there was also activity by carnivores in the cave? So is it cases of maybe like Ice Age hyenas bringing remains into a den and then they got buried there? Do you know how or have a hypothesis how the bones ended up there?
We are working on this in detail. Honestly, my impression, because we have so few fragments, and they're all like we have no fragments except for one of the cranial fragments that are freshly broken. We don't have any two fragments that come from the same individual. You have one tooth here, one finger bone there. We have quite a few of the hominin fragments. For example, the Denisova 11, the hybrid. Is clearly digested. The bone shows surface damage that fits with, stomach acid. There's also a finger bone, a Neanderthal finger bone, Denisova 9, that is also digested. It contains actually more hyena DNA than human DNA, which doesn't indicate that it's a hyena-human hybrid. It's likely simply that it's been digested by a hyena. Hyenas, they eat bones and then they throw them up again. I think that happened to most of these individuals. Yeah, it's different in some of the other sites, but in Denisova at least, that's how that's I think what is the best explanation.
Yeah, because I think for other like archaic humans, I think the caves were not always used as burial chambers. They're more like they think cultural sites where they go and do some kind of rituals. But I don't think that's where they bury their dead. Or am I right or am I wrong on this?
I mean, I'm not completely convinced that that Homo naledi actually that is really a burial chamber. I think the that is still very much of an active question, but it's clear that some others, Homo heidelbergensis, or whatever they are, early Neanderthals from Cima de los Huesos in Spain, seem to have disposed of their dead in a cave, just thrown them into a deep pit. Um, I think it is a possibility, but yeah, it's going to be hard to test until we find more material and in a with a better context. A lot of the Denisovan material, especially the finds we have that are identified via zooms, via this collagen fingerprinting, we only know the square meter they come from and the layer, and not in more detail. So it's hard to reconstruct things.
Okay. Maybe one more question before we move in from questions from the audience. I'm just curious, why do you think Denisovan bones are so rare? Is it just that we haven't looked at the right places, or is it, as you mentioned, maybe some material has been misidentified as being a different human species, but in reality they are Denisovans? We just have the material, we just haven't recognized that they are Denisovans.
It's probably a bit of both. For example, we have so many Neanderthals because there's been so much paleoanthropological research in Europe and the Near East. Asia is still much less explored, especially relative to the size, of how large these regions are. There's been much less research, and so I think we found less due to that. But also, as you said, I think there is a lot of material that has simply not been identified as Denisovan. There's a bunch of material from China from the last 300,000 years that potentially could be Denisovans, and I'm pretty sure that several of them actually are Denisovans.
So in that case, would the name Denisovan? Well, I'm not sure if there's a scientific name. Is it Homo Denisova or something? Which one would have seniority? Would we have to sing
Yeah,
Denisovans to use another name that's been used before, or
I
how would
mean,
that work?
Chris Stringer, who is actually one of the people who described the Harbin cranium, who's in the chat, he said that he refers to it as Homo daliensis, which I think is correct. So there's a cranium from a site called Dali in China that might belong to the same population, and I think it was named in the 70s, so it would be the taxonomically correct name then would be calling it Homo daliensis. But I think the Denisovans as a common name will probably stay around for a while, because
Because
it's
I think erecting
catchy.
a Homo species name just based on that little phalanx from Denisova, I think that would pushing the limit even for paleoanthropology, where you're used to dealing with fragmentary remains. That would be pushing the limit to make a holotype out of that
Yeah,
little.
I mean, we did actually the very first paper on the finger bone did propose a species name, Homo altaiensis,
Oh,
but
okay.
the reviewers at Nature thought were of the same opinion that naming a species based on the finger bone might be a little bit overdoing it,
Yeah.
and so we didn't do it. Um, but yeah, I think the senior synonym is Homo daliensis, and then probably we should go with that if you want to use taxonomic names.
Awesome. Cool. Thank you. Well, we have a few questions coming in from the audience. If you're okay with answering a few of them.
Yeah, of course.
Uh, I'll read the first one. Okay, a question from Robert. He's asking, how is damage to DNA? How is the damage to DNA a function as a function of age calibrated? So basically, how, because I think, yeah, you've shown that with time there's more
Yeah,
damage, and you can identify how old the DNA is. So,
it's very approximate because it depends on much more than just the age. It also depends on things like the temperatures that the bone is under likely the chemical environment, the amount of humidity, and so on. But there are studies that show that if you have specimens that are less than 50 to 100 years, they usually show few of these damages at the ends of the fragments. By a few thousand years, you have it in 10% of the cases. So it's approximate, but usually what people do is simply that if they really want to ascertain that DNA is old, they only use the fragments that actually show these characteristic damage patterns. And any fragments that don't show these damage patterns are assumed to be contamination, which is the conservative approach. You likely lose a few old fragments, but you probably manage to exclude most of the contamination.
Have another question. It's from Anne, who's asking, how many generations can the DNA of either Denisovan or Neanderthal be carried? Obviously, none of these hominids are around, and yet the DNA still persists.
The DNA remains in there. It gets selected out a little bit. What happens with it, what is the bigger problem is that the fragments get smaller and smaller and smaller, and after a while, you just can't recognize them anymore. So, if you think about it, the first individual who was a hybrid of modern humans and Neanderthals had one copy of each chromosome that was Neanderthal and the other copy that was a modern human. When that individual had children, you had meiosis, you had a crossing over occur, so recombination, where the paternal and maternal chromosomes of this individual, so the Neanderthal and modern human-like, got recombined. And so the kid of this individual got chromosomes where part of it was Neanderthal, part of it was modern human. And in the kid, it would be roughly half-half. So you had large segments that were Neanderthal-like. In the next generation, some of those segments again got cut up by recombination. So you can actually use the length of these segments to tell how far back these recombination happened. And so it will, the Neanderthal DNA will stay in us. It'll just be smaller and smaller fragments, and at some point you won't recognize it anymore. But it does stay in, which is pretty cool.
Cool, thank you. Oh, I see we have a comment from Chris Stringer, paleoanthropologist at the Natural History Museum in London, making comment about the Harbin cranium that he refers to, Homo daliensis, that we talked about earlier. He said that anyone interested can type Chris Stringer Harbin on YouTube, and there should be a video to give the audience more information about that. So thank you for the information. We'll definitely look into it. We have more questions coming in. Question from Aidan. What is your favorite and why? I presume it's probably related to Denisova, but let's open it up. Favorite specimen?
It's kind of hard. No, I definitely like the Denisova 4. That tooth is probably one of my favorites. But before I worked in Uzbekistan, and we found parts of a Neanderthal child cranium at a site called Obi Rahmat, and I spent months with those fragments. So probably Obi Rahmat is my favorite. It's not much. It's only 6 teeth and about various little fragments of the cranium, but they are very close to my heart.
Cool. Yeah, I think we all have one favorite specimen. It's not necessarily the most beautiful, but sometimes there's something special about it that makes it a favorite. Yeah. We have a question now from Tracy, who's asking if you can recommend any books that would provide a good summary about modern origins. And don't feel free to do self-promotion if you have your own book that you want to
I haven't written a book about that, but I would actually recommend one of the books of Chris Stringer, who commented above. I actually don't remember what the title of Chris's book is, but it's very good. And if you're more interested in the genetic things, there's actually there's a good book by Tom. What's his last name? Oh my God. I will remember in a moment. There's a good book on Denisovans and ancient DNA research that came out recently as well.
Yeah, it's such a popular topic. Feels like almost once or twice a year there's a new paper coming out, so there's always new developments. Yeah. So I'm sure if you go to Chapters or Indigo or any bookstore, type human origins, there's going to be lots of options out there. But one by Chris Stringer is a good overview. Here we have a question by Larry, who's asking, do you have any idea of where and why the Neanderthal and Denisovan lineages separated? I think you showed that was it 650,000 years ago? I think that
depending on which data we use, somewhere between 600 and 400,000 years ago. I think it was simply geographic. I think that this population left Africa, and some of them migrated towards the east, some of them towards the west, and they were just geographically far enough apart that they weren't in genetic connection anymore. At least the ones we have, at the extremes. I mean, the problem is, for example, as I mentioned, DNA preservation depends very strongly on the temperature. And so we don't have much ancient DNA from Africa or the Near East, or also from Southeast Asia, sadly. And so one of the interesting questions is, did we have, were there populations in the Near East, for example, that were actually where Neanderthals and Denisovans still kind of were in contact? Would we have populations there that are more intermediate between these two? But without DNA, it's going to be very hard to figure it out. But yeah, I think it was simply geographic separation.
Cool. We have a question from Rodrigo. His question is a little tangential to what you talked, but he's wondering if you could provide some input on that. It says 10 years ago, researchers thought that language appeared in Homo sapiens some 100,000 years ago. Even more, some of them thought that a genetic mutation happened in Homo sapiens that allowed the emergence of language. However, mainly after the discovery of Denisovans and the sapiens Neanderthals Denisovans interbreeding, some researchers have put forth the hypothesis that these three groups were able to communicate, pointing to the fact that language at least had begun to appear before the separation of these three groups, perhaps 1 million years ago. Do you know something about that topic?
Yeah. I mean, I was always very skeptical of this idea that there's a single genetic mutation, kind of a magic spark that changed everything, that led to the evolution of language. I always was very skeptical of this, especially because in the end, if you compare the genomes of Neanderthals and modern humans, for example, or of Denisovans and modern humans, there's actually very few positions where 100% of recent humans are different than the Neanderthals or the Denisovans. There's actually very positions that are within genes that actually change the protein. There's only a handful, maybe 100 and something, and I just don't think that is a far too simplistic explanation. Genetics and genes work in a much more complicated way. I do think that the origins of language lie likely much further back, but of course this is very hard to test. But honestly, in my opinion, many of the behaviors that we know that Neanderthals engaged in, the production of personal ornaments, the use of pigments, and so on, are things that are usually referred to as are things that are hard to imagine without any kind of language. I am pretty sure that Neanderthals had some kind of language. Now, the fact that these groups could communicate, I mean, if you take me and put me somewhere in even in East Asia, I'll have a very hard time conversing with the people. I mean, we all speak languages, but we speak different languages. Neanderthals, Denisovans, and modern humans likely would have spoken different languages as well. None of them probably related to any language that is around today. But I think I'm sure they would have been able to communicate somehow. In the worst case, non-verbally. I think it's relatively hard to study this. Language doesn't fossilize. The parts of the body that are the most important for language, like the brain, don't fossilize either, and so it's always going to be hard to study this in detail. Oh yeah. Anadolu is right. The book that I meant was Tom Hyam's book. Yeah, it's The World Before Us.
Oh, good. Yeah, that was the next question. So, someone went on Google found it. Yeah, to come back to language, I think. Well, correct me if I'm wrong, but I think part of the debate as to whether or not Neanderthals could speak was the fact that the hyoid bone, the bone on which the tongue attached, was poorly known. But then since then, I think they've discovered some, and I think there was even a
Yeah.
finite element analysis on the hyoid bone of a Neanderthal that showed that, yeah, pretty much it would have been built up to do what it's supposed to do.
Yeah. And you know, and hyoids in modern humans actually vary quite a bit. So the we have a single hyoid from Kebara cave in Israel for a Neanderthal, and that, you know, would fall within the modern human range of variation, I think. Yeah, it's going to be hard, but of course these are the interesting questions. I mean, for me also the question is, you know, how did, for example, you know, with this Neanderthal Denisovan hybrid, how did this actually happen? Was this just, you know, a Neanderthal woman accidentally met a Denisovan man in the forest, or were these groups interacting routinely? It's going to be very hard to figure this out, but of course these are the most interesting questions.
Yeah. Cool. Well, I have maybe one last question before we let you go, because I think we're done answering questions from the public. My question is about that last bone you show the mandible from Tibet. That basically seems really well to me. I'm used to looking at maybe modern human jaws, but I'm not familiar with ancient humans. But that jaw looked so massive and so short. Is that typical for archaic humans, or would that be totally? That's what you said is totally atypical for Neanderthals and humans. It has to be a different species.
No, I mean, of course, compared to a modern human, it's very robust. It's much less, you know, surprising if you compare it to things like Homo erectus or so. It fits with that. It's fascinating. There's a mandible that's very similar to this mandible from Tibet that was found in the Strait of Panghu between Taiwan and mainland China. It was dredged out by fishermen at some point. And so again, there's absolutely no context, but morphologically, it looks very similar to this jaw from Tibet, and so that might be another Denisovan as well. So yeah, it is much more robust than modern humans. I mean, one of the interesting questions for me too is also with the Harbin cranium being so large is how large these individuals were. They were likely quite large bodied. And of course, we don't know. You know, the finger bone is not really ideal for doing any kind of body mass estimation. But I think the dental and cranial material indicates that they were relatively massive guys. I imagine them a bit like, you know, American football players in stature. But you know, maybe I'm wrong. We'll see. Hopefully, one day we'll have a more complete skeleton and we can tell a bit more.
Yeah, well, let's hope. Yeah, the rate at which discoveries are being made, yeah, we may not have to wait that long. Hopefully, the pandemic ends and you're allowed to go back in the field and make new discoveries. That hopefully we won't have to wait too long for exciting new discoveries.
Yeah.
Okay, well, awesome. Well, I think that was all the questions we received. I've usually seen the Q&A. You see lots of thank yous for the great talk. So yeah, I would like also to thank you, Bence, for a very fascinating talk. It basically makes us dinosaur paleontologists very jealous of things you can do that you will never will be able to. So thanks again, for a great and fascinating talk. And also I'd like to thank everyone in the audience who asked questions and have joined us to listen to the talk. So hopefully you enjoyed it, and you'll join us next week for another talk, as we have talks every week. So next week, guest speaker will be Dr. Garrett Hopkins from Western Oregon University, and we'll be talking about salty salamanders and bright bugs, an evolutionary perspective of life in stressful environments. So talking about a little bit conservation, but also a little bit about evolution of among salamanders and amphibians in general. So I invite you to come back next week to listen to that talk. And again, like to extend our sincere thanks to Dr. Bence Viola from the University of Toronto for a great talk today. And hopefully, next time you're in the Alberta, the Drumheller area, if you come to the museum, definitely let us know, and we'll try to get together and maybe show you behind the scenes, see what's happening at the museum.
Yeah, that would be great. Thank you very much. And thanks, of course, all of you also for both for attending and for the questions. Thank you.
Awesome. Well, thank you so much. And I'll see you hopefully next week.
Yeah. Goodbye.
Thanks, Vance. Bye.